and I'm thrilled to host the second installation of the Technology Roadshow today. As some of you may recall, we had our first roadshow earlier this year in May. Now, before we get started, just some quick housekeeping. This session will be recorded, and by participating, you consent to being recorded. This recording, presentation slides, and Q&A transcript will be made available to all registered attendees after the event. The chat box is open for your questions, and should we run out of time, we will follow up offline and share the transcript afterwards. To ensure we capture all your questions, please take care to select to everyone when you type your question. Now, let me set the stage by sharing a quick background for context. PUB, via administration of RIE funds from NRF, has been actively investing in R&D since 2002, most notably leading to the launch of our third national tap, Newater, in 2003. Since then, PUB has administered over 710 projects, totaling $813 million, in collaboration with partners from 27 countries. And 1 in 2 of these projects progresses to the next phase or are successfully implemented. PUB's R&D approach consists of 3 levels and spans across 9 technology domains. Today's lineup will focus on the latest research and innovations from EWTCOI, NUS, and NTU, targeted at water treatment, desalination, and reuse. Next slide, please. In this domain, the 3 key challenges that researchers are attempting to solve are: 1, how to bring down the energy costs from desalination; 2, how to increase new water recovery efficiencies; and 3, how to ensure safe water is consistently produced despite the unpredictability of the source. Next slide, please. Membrane technology are an integral component of water treatment, desalination, and reuse. Membranes act as a filter to separate contaminants from water, or in the case of desalination, they are used to extract fresh water from seawater by removing salts and other solutes. SGMEM was set up as a national one-stop hub to bring together the players across the membrane ecosystem to accelerate the lab-to-industry translation of research and inventions towards solving actual real-life needs and applications. Now, without further ado, I would like to invite Mr. Ethan Siu, the Innovation Manager of Food, Health and Sustainability at the Innovation Partner for Impact, IPI Singapore, to kickstart the session by giving us an overview of the membrane technology landscape. Ethan, please.
Thank you, Joanne. Good afternoon, everyone. I'm Ethan from IPI. Today, I will be briefly sharing some insights on the current membrane technology landscape. But before that, I'll do a brief introduction on IPI and our programs to help companies leverage on innovation as a business strategy. There are some useful QR codes and links throughout the presentation. Feel free to scan them and access the resources accordingly. IPI is set up in 2011 by the Singapore government as a neutral intermediary to catalyze innovation in Singapore enterprises. It is actually our 10-year anniversary this year, and over the years, IPI has been supporting Singapore companies by facilitating connections with the innovation ecosystem partners and other co-innovation avenues. By accessing to these external resources, companies are able to develop new solutions, solve problems faster, at a lower cost and risk. Here is a snapshot of IPI's innovation services. The first pillar here on open innovation services is where we engage companies one-on-one through modes such as technology scouting, and we also carry out technology landscape studies on particular topics of interest. And companies at a certain growth stage may also benefit from strategic advisors, and we do that through the Innovation Advisory Program. And we also have an innovation marketplace. That's where you can find curated tech offers, problem statements for you to look through. And if they're relevant to your needs, you can reach out and we'll be happy to facilitate the connection. And on the events front, our annual flagship technology industry brokerage event, Tech Innovation, which concluded recently, is where we foster collaboration between the international and local tech providers and seekers. Our technology forums is a platform where we share insights and knowledge, and also emerging technologies that can address the needs of the future and creating opportunities for companies to tap on. All these are examples and platforms that IPI readily shares knowledge and insights to explore new technologies and business opportunities. In the next few slides, I will elaborate more about some of our services. Technology Scouting is a service where IPI supports companies to actively seek out collaborators and partners with the aim to speed up projects, plug development gaps, and possibly explore licensing. Over the years, we have actually developed strong relationship and trust with some of our clients. And this also means that we function as their external tech scout, and we keep them abreast of interesting technologies. Technology landscape studies is where we help companies deep dive to certain topics and uncover opportunities or competition. And this actually helps them in their strategic and investment decisions. So increasingly, corporates and government agencies, even with the likes of PUB, also see the value of crowdsourcing for solution providers to their problem statements. And IPI provides the platform for these innovation challenges, so that the end goal is that, driven by the needs-driven problem statements, good partnerships and commercial outcomes can be derived between the participants and the solution providers. Some of you may be familiar with the Innovation Marketplace. It is a repository of tech offers and needs from a wide spectrum of industries, domains, and postings that are curated by IPI. They can come from either local or overseas parties who are seeking potential collaborations. So if you are the tech owner and looking to monetize your IP, this may be a useful platform for you. I'm also sharing the Water Technology and Singapore Membrane Consortium Technology Bundle here as an example. Feel free to scan the QR codes to browse the tech offers. So through events, IPI frequently shares market and technology insights. And usually this is on emerging topics such as new technologies to deal with difficult to treat industrial wastewater. We also publish white papers that are free for download from past forums. Feel free to go through the links and scan the QR codes to view the past forum recordings and download the white papers. So from these events and publications, we highlight business and possible co-development opportunities. And these are partnerships that IPI would like to facilitate. Lastly, I would like to invite you to subscribe to IPI's Tech Alert to get the latest technology and event updates. For the next part of the presentation, I'll be sharing with you some insights on the current membrane technology landscape. Before we go into the technology landscape itself, it is always interesting to take a look at the commercial outlook of the technology as a whole. The membrane technology market stands at around $6.9 billion in 2021 and is expected to grow to $9 billion in 2027 with a CAGR of around 4.2%. The four main sectors in the membrane market includes industrial, medical, wastewater treatment, and water treatment, of which wastewater treatment segment is expected to be one of the fastest growing with a CAGR of 5.3%. Through the analysis of the membrane technology patents for the last 10 years, we found that there are 9,096 patent applications worldwide. The top countries and regions filing for membrane technology patents are US, China, European Union, Canada, Australia, indicating that these are key markets, of which US takes the lion's share with close to 28% of all the patents filed. If we go another level deeper, we can look at IPC codes. IPC, or International Patent Classification, is a method used to classify patents according to the areas of the technology they are pertaining to. Here, we look at the top 5 IPC codes relating to all membrane technology patents in the last 10 years. Beyond applications in water and wastewater, membrane technology is also frequently used as a separation technology in food, chemicals, and biomedical industry. Next, let us zoom in to take a look at the main topic that will be covered by the rest of our 3 speakers, nano composite membranes. I think most of you here are familiar with LG Chem's nano H2O nano composite membranes, invented by Dr. Eric Hoek and his team. The nano H2O membranes incorporate zeolites as nanofillers into the polyamide active layer, effectively improving the flux and rejection properties. So in general, nanofillers can be classified as either solid fillers such as silica, titanium oxide, and graphene oxide, or the other category of fillers, which are porous fillers, which includes inorganic materials like morphs, zeolites, and also organic fillers like water channel protein, aquaporin. There has also been a lot of interest in completely synthetic channels, known as artificial water channels, addressing some of the cost and stability challenges with the aquaporin molecule. Let us take a closer look at graphene oxide membranes. There has been 154 patent applications filed on graphene oxide membranes for the last 10 years. If we zoom in and look at the top 5 assignees in terms of the numbers of patents, EWTCOI, our next speaker, stands out at number 4, tied with Halliburton. EWTCOI will be sharing more about their technology on graphene oxide production and incorporation into the UF and NF membranes. Next, let us zoom in to look at artificial water channels, or AWCs. As the area of research is relatively new, it will make more sense to look at journal publications instead of patents. Since the first identifications of AWCs by Leduc in 2011, we have seen an increasing research interest over the last 10 years. So notably, number of journal publications in the last 4 years have gone up significantly. Our second speaker, Prof. Prakash Kumar from NUS, will be touching on photomias, the type of synthetic channels, and our last speaker, Prof. Wang Rong from NTU, will be sharing with us her work on peptide-based PRPH channels. I've come to the end of my presentation. My contact details are here on this slide, and I'd like to thank PUB for giving me this opportunity to share about IPI services and a brief analysis of the membrane technology landscape with all of you today. Thank you, and hope you enjoy the rest of the roadshow.
Thank you very much, Ethan. So as he's given us a snippet earlier, our next presentation will focus on graphene oxide ultrafiltration and nanofiltration membranes for water treatment and desalination. Graphene-based membranes are gaining traction due to their high surface area, mechanical strength, and chemical stability, as well as possessing antimicrobial properties that could help reduce fouling. I'm pleased to introduce Dr. Goh Shu Wen, an R&D scientist at the Environmental and Water Technology Center of Innovation, EWTCOI, at Nyon Polytechnic, where she provides technology consultancy for SMEs in the areas of membrane and water innovations. Prior to EWTCOI, Dr. Goh spent 5 years as a research fellow at the Singapore Membrane Technology Center, SMTC Newry, where she gained extensive R&D experience in industrial applications targeting membranes for wastewater and water technologies. Dr. Goh, all yours.
Thank you, Joanne. Can you hear me?
Yes.
Yeah, okay. Thanks everyone for joining us today. So I have 27 slides, so I will be going really fast. So there are like slide numbers on the bottom right corner of the slides, so you can refer to them later. So let's go on to the next slide, please. So essentially, what is EWTCOI? We are a translational center that is located right in Nyon Polytechnic. So we aim to provide help the industry translate their technology from lower to higher TRL so that they can reach commercialization faster. Next slide, please. So this is a simple snapshot of the services that we provide here. Basically, we have consultancy, contract research, standard service packages, as well as facilities rental. So if you need someone to help you carry out your research or you need some type of testing or characterization, you can look for us. So next slide, please. So today we'll be presenting 3 technologies associated with graphene oxide. I'll go through a brief background first, then before I introduce the first technology. So next slide, please. We started working on the graphene-based membrane around 2011, 2012. That's around 10 years ago. So that was because development and application of graphene in various areas such as battery sensors and membranes were a very hot topic. So there's one particular review by Mary Teresa et al. It highlights the potential performance enhancement and commercial viability of new materials, especially the bio-inspired membranes. So this covered aquaporin, graphene, and CNT-based membranes. So next slide, please. So another review also mentioned that the aquaporin could potentially improve the permeability of membranes by 100 times, and hydroxylated graphene is even higher, like almost 1,000 times. So at the same time, there was a PUB grant called Untitled Channel. So we managed to get an award and we started working on the graphene-based membrane. Next slide, please. So when we start working on the graphene oxide or GO for short, we realized that GO is really very expensive and it's hard to justify for such an expensive membrane in water application. So we started looking for a new method to produce the GO at a low price. So from this table, you can see a summary. It's like the conventional method of geosynthesis are the harmless method and its improved or modified form. So these methods generally require a very long oxidation time. It can be 18 hours. So this could be one of the reasons for the higher cost for production. In our method, we managed to shorten the oxidation time to just an hour. So this will help to reduce the cost for geosynthesis significantly. So next slide, please. So in this slide, the green bars actually represent the iteration of our geosynthesis method. So as you can see, we tried quite a number of conditions to optimize the method to get a GO with a reasonably higher carboxylic acid group concentration and the lighter brown color that we associate with a better GO. So you can see the optimized method is actually the GO 100 degrees. That is the fifth brown thing from the left. Yeah, so it's almost in the middle. So that is our optimized method. Next slide, please. So in the end, we have a semi-automated GO production system. You can see in the picture on the left. The capacity is about 100 grams per batch. So our original intention for having this was to produce the GO so that we can reduce the cost of our membrane. But we also realized that GO has like huge application beyond membrane. So we also welcome industry partner to contact us. You can look at the feasibility of working with us to use this system for your GO synthesis in areas beyond membrane, for example, sensors, battery, textiles, and so on. And next slide, please. So we have come to our second technology. The second technology is about functionalized graphene-based ultrafiltration or UF membrane. Next slide, please. So in this slide, you can see the chemical formula is the structure of the functionalized graphene oxide or GO that we have developed. Usually, GO-UF membrane is made by blending the GO into the polymer dough. However, because we see GO as a type of nanoparticle and our applications are usually in the water or food processing applications, so we need to make sure that the GO do not leach out from the membrane matrix during the filtration process. And to do so, we chemically bond the GO to a polymer backbone and we make it into a polymer composite, which is the black powder that you see in the middle of the slide. And using the black powder, we make it into a hydrophilic flat sheet membrane. There was a video here, but you can't see it because of time limit. Yeah, but you can see the video. We drop a droplet of water on it and then it's immediately absorbed by the membrane, which just shows how hydrophilic it is. Okay, next slide, please. So after the flat sheet, we developed the hollow fiber version. And since this was a collaboration with PUB, we got the permission to collect secondary wastewater from the Crunchy New Water Factory, or KNF for short. The VT was like, of the feed was like 7 NTU, and then the TDS was over 200 ppm, and TOC was about 30 ppm. We conducted a lab study using the system that you see right here. Is a 1 meter cube per day semi-pilot system. And next slide, please. Yeah, this is the result. So to compare the performance of the graphene-based UF membrane, you look at the top chart. So the top chart is a 2 days continuous test run at a constant flux of 50 LMH and 85% recovery. The brown graph is for the control PES membrane, which doesn't have any GO. It operates at a higher pressure above 0.9 bar. Whereas in the same top graph, you see a blue color graph. That's our graphene oxide based membrane. So it operates at a lower pressure as compared to the control at around 0.59 to 0.6 bar continuously without any significant increase in TMP. And the permeate quality was well within the quality required for downstream RO process. So when we presented this to PUB, we were asked to run our GO membrane for a longer duration under the same condition that was used by the KNF factory. Yeah, there is a constant flux of 40 LMH and 90% recovery. So in the second graph at the bottom, you can see the results for over 1,200 hours of operation. Our membrane can operate a stable operation of around 0.4 bar without a significant TMP increase. And the permeate quality is again within the quality required for RO. So this TMP was then about half of what was observed at the PUB plant, which was using other commercial membranes. So the results was very promising, and we started to scale up our system further. Next slide, please. So this is the image of our scale-up system. It's about 25 to 50 meter cube per day, and then it was housed at the PUB Tuas R&D facility, which is for seawater pre-treatment. Next slide, please. Yeah, and we also scale our module to 4-inch. So the gray module in the middle is actually the graphene membrane, and the one on the right is the commercial PES-UF membrane. So we actually bought commercial membrane in its module, but to make sure that we are comparing the two types of membrane side by side, we actually reported it. So both of them are like plotted in the same way, in the same type of module for a better comparison. Next slide, please. This is the TMP comparison for 30 days operation at the PUB site using the seawater. So there was some interruption due to on-site maintenance and shutdown, etc. But in general, I guess you can say that the difference is not as significant as before, because one, we are using a different type of feed water this time, is seawater this time, and secondly, there's also the issue of using a lower amount of GO. Yeah, this is because of a dispersion issue that we face when we scale out the fabrication. So we have to use a lower concentration of GO in this batch of membranes. So we are currently working with a local industry partner on a non-exclusive basis to overcome this dispersion issue. But if any one of you have, like, let's say, a different solution for dispersion and you would like to collaborate with us, please contact us. Our contact at the end of the emails. So next slide, please. In summary, this technology features a first of its kind geomembrane with high permeability and lower operating pressure. So it could be used as a pre-treatment to RO, protein separation, and so on. We are looking for industry partner who can scale up the membrane and bring it further, like, and etc., like for other application and so on. So next slide, please. Yeah, this is the final technology that I'm presenting today. Is on a functionalized graphene-based thin film composite for nanofiltration and reverse osmosis. So the previous technology was on UF, this is on NF and RO. Next slide, please. To form the polyamide selective layer on the NF-RO membrane, typically we use a process called interfacial polymerization. It involves a water-soluble diamine such as the M-phenylalanine diamine and an acid chloride monomer such as the trimethyl chloride. So the linkages in the polyamide TFC layer in the NF-RO can be deactivated with repeated chlorine amine dosing, resulting in a reduced rejection over time. Hence, we propose the idea of using a functionalized membrane, functionalized graphene, which is the amine acid functionalized graphene that you see in the picture here, and the acid chloride functionalized graphene. So two of them react together to create a geo-based TFC layer. Next slide, please. There was an animation here, but it's gone. So the initial hypothesis was that the two types of functionalized graphene sheet will react each other, and then in the end, they will form this continuous thin sheet with a tunable channel and pore size. Next slide, please. So these are some of the SEM images of the conventional TFC and the graphene oxide-based TFC. On the left is the conventional TFC, and you can see that the surface is relatively rough. So typically, a rougher surface is more susceptible to fouling. On the other hand, the surface of our GO TFC is very, very smooth. So we realized that we can even reduce the roughness further when we reduce the thickness of the GO TFC layer. Next slide, please. So to evaluate the performance of our GO membrane, we select two GO TFC of different thickness. As you can see from the table, the top thinner TFC layer is the flux for it is higher at almost 67 LNH per bar, but sodium sulfate rejection is lower at 75%. On the other hand, the flux for the thicker TFC layer is lower at around 11 LNH per bar, but the sodium sulfate rejection is higher at 91%. So by tuning the TFC layer thickness, we could tune the membrane flux and rejection property according to the application it was meant for. Okay, next slide, please. And that was the result for the flat sheet. After that, we developed the coating method for hollow fiber using the G support, which is a ultrafiltration membrane. And then batch 1 to 5 were membranes that we coated using different methods. We compared the results with the literature reported membrane and commercial GE and Dow membrane. So if you can see from the stars up there is like the membrane performance is actually comparable to the NF270, but a major challenge that we face is the defect that form as we develop the TFC layer. So as you can see earlier, it's like the orientation of the geo as it comes together to form the TFC layer is very important. So sometimes if it's not orientated or there's some misalignment, it may result in some defects. So these minor defects actually reduce the salt rejection. So that was a very that was a major challenge that we encountered. But in the next slide, please. Sorry, next slide, please. Oh, are we at the next slide? I think my computer is hanging. I assume we are at the next slide. So we found that despite the small defects, the same membrane can be applied for divalent ions rejection or diffractionation. So if you see in the picture, there's actually we are using a red color dye. So there was a video, and then with the red color dye is at the shell side of the membrane, and then the lumen side is the permeate. The permeate is actually clear. Yeah, I think there's something wrong with my internet, but I hope you are hearing me fine.
Yes, we can hear you fine, and
Yeah,
you're
sorry.
on the right slide, so no problem.
Okay. I'll just continue talking. So
Sure.
yeah, we managed to achieve like 99.6% dye rejection using the same type of membrane that doesn't have a very high salt rejection. So the same membrane can still be used for some application. Next slide, please. So in summary for this particular technology, the key feature is that the functionalized geo-based TFC layer could be coated on both flat sheet and hollow fiber. So you could use it for applications such as dye salt fractionation or dye separation. So we are looking for industry partners who can collaborate with us to work on the defect and bring this technology to the next level. Next slide, please. In summary, we presented 3 technology today. So firstly, the semi-automated geo-protection system with a capacity of 100 grams per batch, where we welcome like industry partner to work with us to use this system for your geosynthesis in areas such as membrane, sensors, battery, textiles, etc. And secondly is the functionalized geo-based UF membrane, the ultrafiltration membrane. It has a lower operational TMP. So we also welcome partners who want to test this.
Okay, I think we may have lost Shuwen. I think she may have dropped off. The good news is this is the last slide, and I think her presentation was really succinct. I presume she will be calling back in. So if you have any questions, please feel free from this presentation, just type it in the chat box and we'll get to it shortly. So I'll proceed with the next presenter. The next one will touch upon synthetic water channels with high ion rejection properties. Research has shown promising results that facilitates the fabrication and scaling up of biomimetic membranes. I am pleased to introduce Professor Prakash Kumar, full professor at the Department of Biological Sciences at NUS. He specializes in the research areas of plant development, tissue culture, plant psychology, and molecular biology, hence this nature-inspired research topic. He's also the editor of Plant Cell Reports, BMC Plant Biology, and Plant Biotechnology Reports. Professor Kumar?
Thank you. Thank you for the kind introduction, and I would like to first of all thank PUB and IPI for providing the platform and giving us this opportunity to present our findings. So as you can see, the title of the talk is Synthetic Water Channels with High Ion Rejection Property. Now, why would a plant biologist work with this? It's, by the way, plant physiology, yeah? Plants don't have psychology. So why would a plant physiologist work with this? So we were initially studying the mechanism of desalination in mangrove plants. So desalination became an aspect that we were studying, and we worked initially with the Aquaporins, and we have successfully completed that project, but I will not be presenting details on that. So the current project, the title of the current project we are working on is at the bottom of the slide, that is design of synthetic channels to regulate flow of water and ions across membranes, right? So the idea is to use the technology towards water purification and biomedical applications. So we are collaborating with a French collaborator, Professor Gilles Guichard at Bordeaux. He also is with the university there, and then with the CNRS. So I have a collaborator, Professor Kinney, and we have 2 research fellows working on it. The next slide will give you a brief technology overview, right? So what we have are called Oligo-Urea foldamers, and I will briefly touch on what these are. Okay, so the top right box tells you that what we have done so far, we have designed, synthesized, and characterized 13 such molecules. Now, why so many? Because these are totally novel products, right? So when we synthesize them, we don't know what a given molecule will do, right? So we have to synthesize several. Now, is that just purely a random synthesis? No, right? So we have synthesized this, I'll tell you in a minute. So we have synthesized these, and we have carried out functional evaluation, and based on that, we have selected 2 water channels, right? So where did the idea come from? Well, like I mentioned, we've been working on the natural water channel, the protein pore, Aquaporin, and based on the structural insights we gained from the pore forming zone within the aquaporin protein, and the middle of the slide shows you the pore structure of H2. What is H2? It is an oligo-urea foldamer synthesized and reported by our collaborator, Gilles Guichard's lab. Now, that is not a water channel, right? They were using this for biomedical applications. They've published it in Nature Chemistry in 2015. So using the 2 pieces of information, we came up with our rational design and synthesized molecules based on the structure of H2. Right? So, and what is an oligo-urea now? Most of us are familiar with what proteins are. If not, they're made up of individual amino acid units joined end to end, right? So like the bottom left shows you, a peptide chain has what we call a peptide bond, right? So that is the CONH bond. Now, an oligo-urea is made up of building blocks that are modified amino acids, right? So what is the modification we introduce? So we introduce the modification such that instead of the normal peptide bond, which is the CONH, so these will join together to form the CONH-NH, like in the middle there with the red rectangular boxes, right? So this is the oligo-urea foldamer. So these are, what are the key features of it? The bottom right, we have the little box showing you. It can generate diverse backbones. And these are different, distinct, divergent from the natural peptide polymers, right? So these are non-natural polymers. They are therefore robust and they're tunable. What do we mean by tunable? Because these are made up of modified amino acid residues, we can modify the sequence in which we synthesize those, right? So we can tune them. They form naturally helical molecules. And that's important for self-assembly, right? So because of the difference in the bonding here, instead of peptide bonds, we have the oligo-urea bonds, like I pointed out, right? They are proteolytically stable. Why? Because proteolysis or digestion of protein is by a protease enzyme, and microbes also produce this. So protease enzyme has evolved to dissolve or digest, rather, break the CONH bond. It cannot digest the CONH-NH, the oligo-urea bond, so easily. And the next slide will show you that our molecules, we tentatively have called them OC for oligo-urea channel, right? So we have OC1 and OC2 that we have shortlisted, right? We have several others. They also allow ions, right, to be transported. Now, the important thing is that these are helical and they can self-assemble based on the parent molecule, right? So we have actually observed that in our molecules with the OC1 and OC2. OC2 data is shown on the top row by native mass spectrometry. We were able to show that it forms heterogeneous mixture of oligomeric species. What does that mean? So they come together. So heterogeneous because they form pentamers, hexamers, etc., all the way to 9-mers, right? Monomers. Now, this is important because as you can see, when they come together, they have the ability or potential to form a pore, right? A nanopore, technically. Right, so the bottom left, we are able to show by circular dichroism or CD spectroscopy, we were able to show that by increasing the concentration of our foldamer, we are able to demonstrate that in the aqueous environment, they are able to self-assemble and form oligomers. So this is another confirmation of it. Lastly, we have physically verified that they form fiber bundles. So if it has to form a channel, it should physically form a bundle, right? So channel is a pore inside that. So indeed, the transmission electron microscopy observation, you can see OC1 and OC2 pictures. They form bundles. Well, good. So they form bundles. Now, is there a channel inside? The next slide will show you that we have actually tested the channel. Next slide, please. We have tested the Yeah, well, I guess you have to take my word for it. We have tested the channel forming function, channel forming property of it. Is the slide going to move? Yes. So, and then the channel should allow water to go through it, right? And whatever else dissolved in it by general definition. So we tested the water permeability through these foldamer channels by embedding them in liposomes, right? So this is one of the standard assays for transmembrane pores. So the foldamer embedded liposomes we were able to show by DLS or dynamic light scattering that the radius increases when the foldamer is incorporated, number one. Number two then is the lower panel. We have carried out water permeability studies, right? Water permeability study was was examined through the stopped flow assay. So again, the foldamers are embedded in the liposomes as shown on the left-hand side, and they exhibit high water permeability. The extreme right graph shows you for OC2 molecule that by changing the lipid to foldamer ratio, we were able to, so progressively increasing the number of foldamer channels incorporated into the liposomes, we were able to elicit significantly high net osmotic permeability. Okay, so that's good. Next slide. So this is good. It allows water to go through. Now, one of the important things we need for water purification purposes is that the channel should have high salt or ion rejection properties. So does our molecule have that? The top panel will show, top row, will show you that the fluorescence assay, HPTS-based fluorescence assay, shows quite clearly that with the dotted red ovals, you will see that OC1 and OC2 have high salt rejection ability. So they only allow 7 to 8%. This is in the liposomes. In the membrane, they'll behave differently, right? So in the liposomes, they're able to reject something like 92 to 93% of sodium chloride and potassium chloride. So when I showed the draft slides to Ethan, and he came back with a question, well, does it allow the divalent ions? So we actually quickly tested magnesium chloride and calcium chloride, and they offer similar rejection of sodium, potassium, calcium, and magnesium. And the bottom picture shows you, bottom graph shows you that the foldamer water channels also reject chloride. Specifically, we tested chloride because there was an assay available for it with the SPQ-based fluorescent quenching assay. And as you can see, OC1 and OC2 show very little transport of chloride into the liposome. So the fluorescence quenching is happening inside, right? And what are the others? OC4, OC3, they are the ones that we have synthesized, but we have not shortlisted because they allow significant amount of ions to go through. So their salt rejection is quite low. Next. So then the task next for us was to carry out test membrane fabrication and see if they will behave in the desirable manner in a membrane, test membrane. The way they behave in a liposome may be different from what they behave in the membrane. So the test membrane work was kindly performed at Professor Wang Rong's lab. We thank them for their kind assistance with this. So from the data provided, you can see on the top table that the membranes where we have incorporated the OC1 or OC2 foldamers show significant enhancement of permeability over the control without the incorporation, while maintaining close to 99% salt rejection rate. Now, the beneficial properties of our foldamers are highlighted in the bullet list on the right-hand side. Right, they are short. They have high water permeability, high salt rejection coupled with that, and sequence-based functional tunability. They're conveniently designed, and you can chemically synthesize these. They are proteolytically stable, and there is potential scalability is should be relatively easy compared to, let's say, Aquaporin, etc. So what does the bottom table show? This is just to illustrate how some of the biomimetic SWRO membranes reported in literature properties are, their water permeability and salt rejection. This is from the recent extensive, exhaustive, and excellent review from Professor Wang Rong's lab, right? You can see that the properties of those membranes are more or less, or our membrane is more or less comparable to that or slightly better in terms of permeability. The next slide will show you a list of, again, this is a table from Prof. Wang's recent review. This is listing or illustrating the state-of-the-art industrial flat sheet SWRO membranes. The point that we would like to draw your attention to is in the second column, that's the water permeability. It's consistently below 2 or at or around. The highest that we can point out there is 2.2 LMH per bar in the top row, right? Or the second row, I should say. All the rest of them are below that. So the point I'm trying to illustrate is that our product is showing better features. Next. So what are the potential applications? Well, naturally, biomimetic membranes for desalination is the first one that will come to our mind, right? So as you can see in the bottom box there are about 20,000 desalination plants operating right now worldwide, right? And they're producing something like 100 million meter cubes per day of water, supplying for about 300 million people's use, right? So from what we can see, the demand is going to increase continually over the next years, right? Coming years. So the second point that we want to highlight is that our molecules probably are usable for forward osmosis, for water removal, purification with lower energy consumption. For example, wastewater treatment or water reuse, as well as food processing. For example, concentrating juice or liquid concentrates, whether it's for food or beverage industries or even biomedical applications. Next. So what is the market trend? I think this audience doesn't need to be, convinced about what's going on with the membrane technology. But the point that I want to highlight is that although the left-hand side of the bar graph shows you that adsorption-based technology is the cheapest, but does that mean then the RO technology is lost the race? No, right? Because the bottom box shows you there that RO can process something like 10 times the volume of water compared to the adsorption-based technology. So it is here to stay. Now, the technological improvements one has to come up with this to improve the performance, right? So the right-hand side, the pie chart shows you various competing technologies for water treatment. I'm not saying our competitors. So these are the different competing technologies that are out there. And what we are contributing is going to be in the biomimetic membrane space, that slice of the pie, along with aquaporins. These are the artificial water channels that will be usable as a good product if a little bit more work goes into it. Next, please. So what are the benefits? Well, we know quite clearly that there is a large unmet need, right? So increasing demands for potable water is illustrated in the graph on the right-hand side, which is taken from one of the reviews, right? So the demand for water across the world is going to go up in the coming decades, right? So the existing technologies are not sufficient. I'm not saying they're not good. They're not sufficient by themselves. We need to improve them, right? With improved properties. So what do we bring to the table? We offer two novel oligourea foldamers with improved properties. And these are convenient and cost-effective. They're chemically synthesized by established procedures, right? And the synthesis and purifications are scalable. They're highly stable, self-assembling helical polymers. Their oligourea bonds are resistant to proteolysis. So this is talking about stability again. And microbial degradation is also not easy because the proteases cannot function to digest the oligourea bonds, right? And because we can synthesize, sequence-based fine-tuning can also be built in. And these are usable for, besides the RO, they can even be used for FO. Therefore, our foldamers offer substantial advantages. Next, this is just to show you a brief comparison of some of the key points, right? So again, Ethan was asking, it might be useful if we can list out some of the comparison, right? So the red is basically the drawbacks or limitations, and how on the right-hand side, oligourea, you can see how these red limitations have been addressed in ours. And the blue, well, basically, those are the natural proteins, and then they have larger footprint. So these molecules address that as well. Okay, so I think that's my last slide. Next. So this is just to show you the pictures of the people on the team, and the project is funded by the NRF through the PUB, Singapore's National Water Agency, under the CRP, Competitive Research Program. Right, and I had forgotten to put in my email ID in there, so I will just post it on the chat box now. Right, of course, NUS, you will be able to look for us by name as well. Thank you very much.
Thank you very much, Prof. Kumar. My apologies just now about the
No worries.
fun whisperer. I was like, that's very interesting. So again, if you have any questions for Prof. Kumar, please post them in the chat. Do select to everyone so we can capture them. I'll just circle back very quickly. We had a question that was directed directly to Dr. Gore. I'll just read it out here, although she has responded, and maybe Dr. Gore, you might want to expand if you feel it's necessary. So the question we had from Dylan is, Are graphene oxide-based membranes intended to replace conventional membranes in water treatment or desalination? If so, what is the commercial viability compared to existing solutions, as these are still relatively nascent?
Yeah, sorry, you all lost me just now. But I was at the second last slide. Yeah. Anyway, it could be a replacement for the conventional membrane because we saw that it can operate at a lower pressure, but still the cost of producing the membrane has to be competitive enough with, let's say, existing membrane. So that is one area that we are still working on. Yeah. So yeah.
Okay. Thank you, Dr. Goh. We have a question from Ting Liang to Prof. Kumar. How does the cost of polymers compare with aquaporin channels?
So I would say at the moment, this is still laboratory scale, right? So the cost for polymers will be more or less comparable at this point, but because it's chemically synthesized, we have not scaled it up, right? So once someone scales it up, the cost will be definitely coming down significantly. So what is the drawback? The limitation at the moment is that the building blocks, these are the modified amino acids, right? So those are the so-called building blocks. We can't use directly from the market. We have to modify them. Now, the thing is that since it is such a product, I mean, how should I call it? It's such a molecule which doesn't have a market right now. There is no industrial producers producing and supplying the building blocks, right? So if a commercial producer takes it up, the cost will come down significantly. It will be just like any peptide biosynthesis. There are a number of small protein molecules, peptides being synthesized. So it'll come down to that level.
Thank you, Prof. Kumar. Is this technology up for licensing?
Okay, so at this point, we are still waiting for the collaborators' input on the IP document. So we have, I mean, again, this is another thing that I must admit, we are nowhere near a product that is waiting to be just adopted. So we still need to develop a product. The scaling up has to be also developed further. But the point is that this is quite promising, and because of the chemical synthesis nature, the cost can be brought down quite significantly, and the IP will be filed through NUS, hopefully within the next several weeks. We are waiting for the input from CNRS because we are working with an RCA, so the IP will be jointly shared between us, but it will be filed through NUS, and then hopefully the licensing arrangements and things can be worked out based on that.
Thank you.
Does that answer? Yeah.
Yeah, Prof. Kumar, you have another questions. They're coming in now. From
okay.
Jing Lei. She's asking, he's asking if the synthetic water channel tech requires large space if we adopt it for industrial use.
So again, this is like I mean, what would be a scaled up production facility look like for the building blocks and for the synthesis of it? So right now, this is synthesized in a laboratory. So it's the quantity that we synthesize is in 10 milligrams kind of things. So it's not like humongous quantities that we are synthesizing. This is done in a laboratory, but with proper scaling up, for which my reference is the regular peptide synthesis labs. They're not humongous facilities, but they will be, fair-sized, industrial synthesis facilities. So peptide synthesis exists, and the chemistry is very similar with the modifications on the starting material for amino acids. They have to be modified. Those are the building blocks. And then they have to be sequentially added together. The synthesis, that sequential addition is very similar to the peptide synthesis. So it will be quite a manageable type of technology, and scaling up is going to be with minor adaptations, I would say.
Excellent.
And the membrane preparation, Prof. Wang Rong's lab had used their existing technology, which they have refined one, and I understand they're in the process of publishing that. So the point again is that it is not a novel engineering that one has to come up with. So technology for which exists, and you can adapt it for this molecule.
Excellent. Thank you very much, Prof. Kumar.
Thank you.
So our final presenter of the day is Prof. Wang Rong, a full professor and President's Chair in Civil and Environmental Engineering at NTU. He is also the Director of the Singapore Membrane Technology Centre, SMTC, at Urea. She has served as an editor of the Journal of Membrane Science since 2014 and is now the Editor-in-Chief from the year 2020. Her accolades include winning the Singapore M&D R&D Award in 2013, receiving the prestigious Prince Sultan bin Abdulaziz International Prize for Water from Saudi Arabia in 2016, being awarded the Nanyang Research Award in 2018, and most recently being listed among the world's top 2% of scientists in their field as part of a global list released by Stanford University. Prof. Wang's topic for today will be on novel synthetic water channels, which can be incorporated into the selective layer of RO membranes to enhance membrane performance for desalination and water reuse. Prof. Wang, please.
Thank you, Joanne, for your kind introduction. A very good afternoon, colleagues. I'm Wang Rong from Singapore Membrane Technology Center, Nanyang Technological University of Singapore. It's my great pleasure to be here with all of you. I would like to thank the event's organizer, PUB, for the invitation. Taking this opportunity, I would like to share with you our work on the synthetic channel-based biomimetic membranes for desalination. Next slide, please. Basically, my talk will cover 5 parts. In the beginning, I will provide a short technology overview, provide some background about our work. Then I will highlight technical features and the specifications for our newly developed membranes incorporated with the water channels. Then you will see the potential application and the market trend and opportunities for our new membranes. Finally, I will briefly summarize the benefits if someone adopts our new membranes for application. Next slide, please. Okay, this project is funded by the PUB. That is to synthesize water channel similar to Aquaporin and incorporate into the channels into the synthetic membranes for real application. This project was completed just very recently, the half year ago. We completed the project. We know the state of art of RO technology for water desalination is a thin film composite membranes incorporated with the Aquaporin. We call the Aquaculturing Based Membranes membranes. Actually, this membranes was invented by us at the Singapore Membrane Technology Center at the Urea NTU. We demonstrate that Aquaculturing-based membranes can increase the water flux, also maintain high soil rejection. Some commercialization activity has been initiated. However, we found out to produce Aquaculturing-based biomimetic membranes, there are some challenges here. The first is there is a limited supply of the high purity aquaporins. The synthesis and the production of aquaporin in large quantity actually involve quite complicated procedures and pretty pretty expensive based on the current procedures. So we think about, can we synthesize chemical compounds that mimic aquaporin structure, have a water channel transportation property? Then we hope we can reduce the cost. At the same time, this channel can be incorporated into the RO thin film to enhance the water permeability, similar to the aquaporin-based membrane. That's our objective. So in our project, the first step is to invent new type of the chemical compounds that provide water channel or property, allow the water to transport. Finally, our group, the chemical chemist, we have several chemists in our group. We collaborate with professors from the chemistry department in NTU. We invent a new type of the water channel compounds. That's the peptide appendix pylophylline, PAP in short. This type of the water channel. For the PRPH, we demonstrate these chemical compounds have good water permeability property. We measure this water channel transportation property by using standardized instrument, stop flow. We see these compounds really have the very good water transportation property. We compare these PRPH channels with the natural water channel, Aquaporin Z. You can see from the table, you can see our synthetic water channel have a similar level of the water transportation like the Aquaporin Z. And it also have a very good salt rejection to sodium chloride. We have a set of the data, characterization data, to confirm this PRPH water transportation property and reject the salts. And this invention has a filed patent, and later we also published the paper in a journal called Swarm. You can find the reference if you are interested in this. Next slide, please. The second step is how to incorporate these water channels into the synthetic thin film to make the membranes like the aquaporin-based membranes. We developed a prototype. Basically, the principle is quite similar to what we did for the aquaporin-based membrane, but we need to adjust some parameter. From the table listed here, we compare our synthetic channel-based membranes with control membrane without the channel, and also compare the performance with aquaporin-based membranes. You can see here in the middle, we see with the proper coordination, with the proper amount of the PRPH channel into the vesicle and incorporate into the polymer, the interfacial polymerization polyamide layer. You can see the water permeability is more than 3.2 LMH per bar, and the salt rejection to the seawater, similar to seawater, 35,000 ppm. The rejection is over 99.2 and close to 99.3. That's a very exciting result. Compared with the control, they are around 25 or 30% enhancement in water permeability, while the salt rejection is in a similar level. Then we compare the water permeability with aquaporin-based membranes. You can see very similar level, and you see the permeability and the salt rejection. That's the last row of this table. And the testing is we use the synthetic seawater to do the testing. So that's under the very high salinity, 35,000 ppm, and the sodium chloride. You see the concentration 35,000 ppm, operating pressure 50 bar. We want to find the application of this type of membrane for seawater desalination. Next slide, please. We know the RO technology, thin film type of the RO technology, is the state of the art, as I mentioned earlier. So the membranes played a very critical role to ensure stable operation with high separation performance. Current conventional thin film RO membranes is made through this interfacial polymerization, form the polyamide active layer. Here, I just let you know, we incorporate the water channel into this polyamide thin film layer. So we specially designed these membranes for seawater RO application. Next slide, please. Seawater desalination actually increased tremendously in the past 20 years because of the water scarcity globally. They have tremendous growth in constructing desalination plant for sustainable water supply. And the seawater RO technology is dominant in the market because it have a better energetics and a low footprint. So you can see how important of this SWRO membrane for seawater desalination. Next slide, please. Now, I'd like to share with you the opportunities in Singapore. Singapore started a desalination journey using reverse osmosis membranes in 2005, when we have the first desal plant that's called Spring Desal plant in 2005. Currently, Singapore have 4 desalination plants using the RO membrane technology. Another new one will be ready very soon. That's in Jurong Island desalination plant, 2021. Today, desalinated water can meet up to 25% of Singapore's current water demand. By 2060, this number will increase to 30%. This 30% refers to our future water demand in Singapore. So you can see the opportunity of the RO membrane technology for seawater desalination in Singapore. Of course, this technology, other countries also adopt the desal using RO membrane technology for water production. Next slide, please. So if our technology was adopted by the industry, what are the benefits you can get? Here, I just list several points that's based on my own understanding. When we make these PR-pH channels, actually we calculate the cost. In Professor Kumar's presentation, he mentioned about the laboratory study for the synthesis of the channel. In our group, we synthesize our channel in the laboratory, but we calculate the chemical used, and we found the chemical used to see the cost is around 30 times cheaper than the Aquaporin in terms of the material used, so much cheaper. Because we use the conventional way to make the membranes, just add this channel. So the increase because of the incorporation of the channel into the membranes is minimal. A very small amount will add into the cost for the RO membrane production. We have some calculation. We have done some calculation. We have the number. So the cost is not a concern. I can assure you. We have the detailed calculation. The second advantage, the benefit I would like to highlight here, by incorporating a PR-pH channel into the thin film, the water permeability increase to 3.21 LMH per bar. As Professor Kumar mentioned in his presentation, current SWRO membranes water permeability is around 1 to 2, as shown in this table, in this figure, on the right-hand corner, you might slide, you can see. Compare with these commercial SWRO membranes, their permeabilities are pretty low, just around 1 to 2. That's the best I list here. Now, our SWRO membrane with incorporation of water channel, permeability increased to 3.2, while the soil rejection over 99%. For seawater desalination, the basic requirement for the SWRO membrane rejection should be over 99% because the salinity of the field water is very high. If the rejection is lower than 99%, actually the produced water quality will not meet the requirement. So you can see we need both requirements. The rejection is high. At the same time, we increase the water permeability as compared with the state-of-the-art commercial SWRO membranes. Someone may say, okay, from 2 you increase to 1. It's not a big deal. However, I would like to let you know that SWRO is operated at a very high pressure, around 50 bar to 80 bar. So the increased water permeability can reduce the energy consumption significantly. That's the benefit I would like to highlight to you. Next slide, please. This is my last slide. I would like to thank my staff, students, and collaborators working together to develop this exciting new technology from the synthetic water channel and the incorporation into the membranes. And funding support from the NRF, from PUB is much appreciated. And this experiment was conducted at the Singapore Membrane Technology Center at the NURI, NTU. And the SMTC was funded, and the NURI was funded by the EDB. So I would like to acknowledge EDB support. Thank you very much. I'm more than happy to answer take your question to answer your question. Thank you.
Thank you very much, Prof. Wang. There's actually a couple of questions really in for you. So I'll read the first one. It's from Dylan. What is the life cycle for the biomimetic membranes under these high pressure conditions, as this will affect the cost-benefit analysis?
Oh, that's a very good question. Actually, for the biomimetic membranes, we conduct some the long-term testing. Long-term testing, the longest time is 700 days. That's a 2 years operation. But beyond this, I don't know. Really, I don't know, because that's really we need to use the real data to demonstrate, right? But so far so good. We run the biomimetic membranes in the lab, and everything using the real seawater, real water. We also have the BWRO membrane, so we use the biomimetic membrane. So far, I think the result is good, but how long you define the long term, really, I don't know, because I haven't run the operation yet.
Sure, thank you for that. There's one more question from Sus K. How stable is the synthetic channel when it is subjected to chemical cleaning?
This is also very relevant questions, and this question is really the concern, actually. And actually, we do the cleaning, use the standard protocol, like the normal, the SWRO membrane to do the cleaning. Physical cleaning followed by the caustic, and then the acid cleaning. So far, it's okay. I think the key is that we embed these channels inside the polymer matrix, inside the polyamide layer. Actually, it was protected by the polyamide layer. Also on top, we have like the PVA coating layer to reduce the fouling. It also helps to enhance the soil rejection, also maintain the stable operation. We have the long-term data. I think for this set of the data for the real sea water, we operate like the nearly 1 month. We have the data.
Thank you, Prof. Wang. And one last question here is from Ramakrishna. How does salinity, pressure, temperature, TOC in feed water affect the performance of PRPH?
And so far, you know, we did not see the negative impact because this is the chemical compounds. Even we do some experiment using the aquaporin before, aquaporin-based membrane. That's a natural protein water channel, right? For aquaporin, we see the temperature and the pressure will not affect our membrane because, as I mentioned, already embedded inside protect. But for this PRPH, these chemical compounds quite stable. I will not say they will influence because of the high salinity and the high pressure or the temperature.
Thank you very much. That's the questions so far that we have. If anyone has any other burning questions, do type them in. Let's see if we can get, oh, that's another one that just came in. From Rahul, what was the salt concentration and pressure gradient used to test your membrane?
Okay, as I mentioned, we use the 35,000 ppm. That's the salting chloride, similar to the seawater, the salt concentration. And the test pressure, I think if you go back to my slide, and the back 2 or 3 slides, the control, another one, next one, yeah, next one here, you can see here, test the pressure is 50 bar. You can see test condition, I put here, phase solution, that contains sodium chloride, 35,000 ppm. The testing pressure 50 bar. We simulate the real operation for the seawater desalination.
Okay, great. I think that will answer Rahul's question. Anything else? I think I have nothing else in the chat questions. So thank you again very much, Prof. Wang. Oh wait, something else just came in. Okay. So I think they are coming. Yeah, maybe I won't push it. There's two questions that just came in. Prof. Wang, what is the size of membrane modules that you have tested so far?
Okay, this is a very good question. And actually, we make our synthetic water channel membranes in flat sheet configuration. We just complete the project. And we can make the membranes in the size like the A4 page size. It's not small coupon, actually consider. This is the A4 size we make in our laboratory. If some industrial partner are interested in this, I'm more than happy to work with you to further scale up. A4 size, the membrane coupon size, the paper, A4 paper size.
There's another question from Santosh. I think it's directed to you as well. How is the physical stability biomimetic membrane when they are pressed by feed spaces at high pressure? Do they have any change in structure when subject to different salinities? Operation, flushing?
Here, I would like to clarify, you mean this biomimetics refer to the Aquaporin based membrane, right? Am I right? Yes, I think so. And for biomimetic membranes, actually, currently we work with starch to scale up our Aquaporin based biomimetic membranes. We already done those stability tests so far in the laboratory. We test using the seawater, also cleaning, using operating
I think versus flushing.
Flushing. And we don't use the back flushing because that's the thin film type, and just the normal flushing. We don't use the back flushing.
Okay. Another question, and what is your take on actual biological fouling on these aquaporin type membranes?
Okay, this is also a good question. And actually, we have the data to test the fouling, to run the fouling, the testing using the real seawater. Our biomimetic Aquaporin based membrane, the top layer, also for the seawater, we have like the PVA coating layer. Actually, it's quite stable. And even the fouling, we do the parallel testing within using another two commercial membrane in the same setup to run in parallel to run the long-term fouling test. Our membrane pretty stable.
Okay. I'm going to take it a bit slow in case there are any last minute questions again. Okay, I think. Okay, that's what we have so far. Again, thank you very much, Prof. Wang Rong. If in case we get cut off, you feel free again to chat and we'll follow it up offline. Thank you.
I'm more than happy to answer the question. You can send an email to me. You can just type my name in the NT website. You can find my email address. Thank you, everyone. Thank you for your attention.
So, on behalf of the organizers, I would like to extend a big thank you to Ethan, to Dr. Gore, Prof. Kumar and definitely Prof. Wang for your presentations, for taking the questions, and again to the audience who joined us today and for your engagement. So as we mentioned at the start, we will take any other questions in case they come in later on. We'll post this, we'll take them offline. We will share the presentation, the recording, and also the contact details of our presenters today will be in those slides. So don't worry, you have all the access to reach out to them. Lastly, before you leave, please do participate in the post-event survey that will pop up as soon as you exit the screen. We appreciate your feedback so we can take that on board for future events. So have a wonderful rest of the day. Stay safe, stay healthy. Thank you, everyone.
Thank you.